Okay. Okay, now we are live. Hello and welcome everybody to a new seminar of Sussex Visions. I am Antonio Hinojosa, a postdoc in Leon Lañado Lab, and I'm working in mouse cortex. This series of seminars are part of the Worldwide Neuro, which is an initiative towards a greener and more accessible kind of talks. So I would like to thank the organizers of this initiative, Tim Bogos and Panos Bosellos for starting it. And just for you to know the format of the talk, it will be around 45, 50 minutes of the talk. Then we will have some questions that you can type in the chat, and I will ask Josh at the end of the talk the questions. And also after we finish the talk, we will have a discussion over Zoom, and so feel free to join to that discussion. Also, I would like to remind you that if you want to see our Sussex Vision talks and don't miss any of them, you can join the channel in YouTube, and this way you will get all of them. So today we have the pleasure of having with us Joshua Sainz. Hello, Josh. And he graduated from Yale in biochemistry and psychology. Then he joined the neurobiology lab of John Hildebrand, where he earned his PhD studying the development of mouse neurons. Then he took a year off from experimental science to advise members of Congress in the USA on scientific and technical issues. But then he went back to experimental science, and he began his postdoctoral research at Harvard with neurobiologist Huel Jackson McMahon, and later on with neuroscientist Zach Hall at the University of California, San Francisco. During this period, he started his research on the mechanisms and molecules that drive the formation of neural connections within the neuronal junction. Sainz accepted then a faculty position in the Department of Physiology at the Washington University School of Medicine in St. Louis, and then there he continued his work identifying and characterizing molecular factors involved in neuromuscular junction formation. It was also then when he contributed to generating a technique for tagging neurons with color-coded fluorescent proteins combinations. So this is the famous rainbow mouse, which has been widely used around the world. Then in 2004, Sainz returned to Harvard University, and where he became the founding director of the Center for Brain Sciences. There, his research transitioned to the analysis of synaptic specificity in the retina. And he's currently a professor of molecular and cell biology at Harvard University. To finish, Sainz, Josh has received many professional honors throughout his career, including he's a member of the National Academy of Sciences and also the American Academy of Art and Sciences. He's also been part of numerous advisory panels like the Wellcome Trust or the Howard Hughes Medical Institute. And he's also received numerous prizes like the Gruber Neuroscience Prize in 2017, or recently the Edward M. Skolnick Prize in Neuroscience. So it's a pleasure to have you with us today, Josh, and we're looking forward to hear your talk.
Okay, thank you very much. Let's see, you tell me whether you're seeing what you should see.
Yes, I can see your presentation.
Okay. Yeah. All right, well, it's a pleasure to almost be with you. I wish I were there in person, but there you have it. So I'm going to talk today about recent work from my lab that relies heavily on single cell high throughput transcriptomics. And because that makes people's eyes glaze over frequently, let me just start by telling you how we got into this. And it started with the interest that Antonio mentioned in synaptic specificity, how it is that axons entering an area of the developing nervous system choose among many potential target neurons the ones on which to make synapses. In some cases, they even choose a particular part of a neuron, a distal dendrite, a cell body, an axon hillock, and so forth. And so close to 20 years ago, actually, we decided to investigate this issue in the retina. This is obviously not an audience that needs me to tell you what the retina is or why it's so wonderful. But very briefly, it is a part of the brain, embryologically and structurally. It's about as complicated as other parts of the brain, and I'll try to give you some reason to believe me on that in a few minutes. But it has a lot of advantages that make it easy to ask detailed questions about development, structure, and function that one might like to ask in the cerebral cortex, but one can't because of technical difficulties. It's obviously been pulled out of the skull, so it's very accessible, comes with its own lens. It has clear borders. Its neurons and synapses are arranged in beautiful layers, as you can see in this picture from Josh Morgan and Rachel Wong. Importantly, there are no long-range inputs, so it is the whole circuit from light at the outset to optic nerve at the end. We were interested in it because there are many types of interneurons that synapse on many types of retinal ganglion cells. The numbers here are the ones that were thought to be correct at the time we started this work. Again, I'll update that soon. But in very brief, you probably know this. Photoreceptors make synapses on interneurons in an outer synaptic layer called the outer plexiform layer. The interneurons, there are three classes, horizontal, bipolar, and amacrine cells, and two of them, bipolar and amacrine cells, make synapses on retinal ganglion cells in an inner synaptic or plexiform layer. And then the ganglion cells tell the rest of the brain about what they've learned. And because there are many types of interneurons making synapses on many types of ganglion cells, each type becomes particularly tuned to some visual feature like motion in one direction or a color contrast or an edge or whatnot. And therefore, what the retina does is to send multiple parallel representations of the world that have been sparsified and tuned to a feature onto the rest of the brain. So for us, the interest was in this inner plexiform layer, where in a really very narrow space, especially when synapses are being made, very specific cohorts of interneurons make synapses on very specific cohorts of retinal ganglion cells. And clearly, it's a short distance phenomenon. So it really is synaptic specificity, not, for example, axon guidance. And so over a period of really mostly about 8 years, we use the retina, mostly based on mouse genetics, to label particular cell types, monitor them, profile them, and then find out what they did in loss or gain of function experiments as measured either morphologically or physiologically. And a group of really wonderful graduate students and postdoctoral fellows, whose names I've listed on the right, this is not all of them, but I would say the main players, came up with evidence that a set of adhesion molecules, recognition molecules like the cadherins and protocadherins, and transcription factors like TBR1, SATB1, FESF1 play key roles in setting up this specificity. So this was all rewarding, but it became clear as we went on that if the field was going to get a really satisfying explanation for how this worked, one was going to need another approach, not the retail approach of getting a transgenic line by hook or by crook and finding out something about the cells that are labeled, but rather getting a really comprehensive and unbiased classification of all of the interneurons and all of the retinal ganglion cells and characterizing what recognition molecules and transcription factors they expressed. And this seemed like an impossible dream for a long time, but in 2015, It became possible thanks to the invention of high throughput single cell RNA sequencing methods. It's such a great idea that it was invented 3 times simultaneously. I put in green the invention by Evan McCosco and Steve McCarroll's lab because we ended up collaborating with Evan in the development of this method, or not really the development, he developed it, but in using the retina to validate it and perfect it. And that provided the way to profile thousands, tens of thousands, now people do hundreds of thousands of individual cells at high enough speed and low enough cost to make it possible to conceive of having really a comprehensive and unbiased categorization and characterization. So I'm not really sure single cell RNA-seq has kind of become an extremely popular and much used method over the past 6 years. But I don't really know whether some of the people in this audience might need an explanation, so I'll try to split the difference and give you a very quick one. So essentially what one does, this is drop-seq, but all 3 methods use the same basic sort of platforms. We actually now use the commercial one called 10x. Probably most of you who do single cell transcriptomics do the same. So in any of them, you dissociate a tissue into individual cells and then isolate the cells. Nowadays, you can isolate the nuclei and then pass them through a fairly simple microfluidic device in which a single cell is encapsulated with a single microparticle in a nanoliter-sized oil droplet. And inside that oil droplet, the cell lyses and messenger RNAs are captured by DNA oligonucleotides that are derivatized to the microparticle. They end in oligo-DT, and therefore they capture polyadenylated messenger RNA. Now then the whole emulsion is broken, and the RNA is turned into DNA, amplified, and then sequenced. And the secret of the method is that you can now capture, amplify, sequence the messenger RNA transcriptome of literally thousands of cells in a single tube rather than needing thousands of tubes and dozens of graduate students. And that's really the key to saving time and money. Now, the DNA oligonucleotides on the microparticle are elaborately barcoded. And so that means that computationally, once you have all the sequence, you can divide the multiple sequences into those that came from cell 1, those that came from cell 2, and all the way up to cell 5,000. So here is a display of the results from Evan's first big experiment on retina, in which he profiled almost 45,000 cells and then computationally grouped them into 39 clusters and displayed them on what's called a t-SNE plot. And so this display has 44,808 dots, and their colors and their grouping represent what the computational algorithm thought were cells that were sufficiently similar to each other in terms of their transcriptome, that is their catalog of messenger RNAs, to be potentially all the same type. It's nominally an unsupervised method, but as we can discuss later, we twirl the dial, so it isn't completely unsupervised, but mostly. So at any rate, because it was the retina and we had so much ground truth, we could look at genes expressed by cells in each of these clusters and ask what they represented. And the good news was that each cluster represented a type or a small group of types that we knew about. Amacrine cells, photoreceptors, bipolars, horizontals, ganglion cells, those are the 5 canonical neuronal classes in the retina, as well as non-neuronal cells, including Mueller glia. And in addition, their transcriptomic relationships to each other, which you can see in the dendrogram on the left, made sense. So rods and cones were each other's closest relatives. All the bipolar cells were each other's closest relatives, and so forth. So that's the good news. The bad news is that there were only 39 clusters, and we already knew that there were more than 39 cell types in the retina. And the problem was that even though this seemed to be a ton of cells, and it was, most of them were rods, and therefore there weren't enough cells in the most heterogeneous classes, bipolars, amacrins, and ganglion cells, to have the computational power to divide them into individual types, and in some cases to even capture the sparse types. And so what we went on and did then was to use methods to enrich ganglion cells, amacrine cells, and bipolar cells separately so that we'd have enough of them to take the job through to completion. And over the next few years, we did that, finding 15 bipolar, 46 ganglion cell, and believe it or not, 63 amacrine cell types. And in each case, we could look for genes that were selectively expressed in individual clusters, putative cell types, and then use morphological methods, immunohistochemistry or in situ hybridization, to see whether the types defined molecularly corresponded to types defined by criteria that neurobiologists would care about, which were morphology and physiology. And the good news is that at least for the retina, we don't know if this is going to be true throughout the nervous system, but at least for the retina, they did. We did a comprehensive match physiology to morphology to molecules for bipolars and spot checked the ganglion cells and amacrine cells. So we ended up then by 2020, a year ago, with what we think is a nearly complete cell atlas of the mouse retina. I say nearly because, of course, there'll be no shock to anybody if one or two more show up. But from everything we've seen so far, it's pretty complete. 130 neuronal cell types and 10 or so, probably more than that, non-neuronal cell types. And that is what makes me really believe that the retina is about as complicated as other parts of the brain. This is a number very similar to what people have found, for example, in visual cortex, motor cortex, and so forth. Interesting, the number doubled over the decade between 2010 and 2020. And there's lots of reasons for that, but I think it's safe to say that the biggest contributor to that increase is the single cell transcriptomic method. So as this atlas neared completion, we, of course, wanted to use it to ask interesting questions about biology. The atlas is an atlas. It's not interesting in and of itself. And so over the past few years, we've been trying to use that atlas as a foundation to learn things about the development of retinal cells, their responses to injury, retinas in humans and non-human primates, cell types in which disease-associated genes are expressed, and finally, evolution of cell types. And so what I'm going to do today is focus on the first, second, and fifth of these, development, selective vulnerability, and evolution, and tell you fairly briefly three stories that are preliminary, at present unpublished, although I'd like to think they're not, in the long run, going to be unpublishable. So let me start with development. Lots of questions there, including the one that set us off in the first place about what recognition molecules and transcription factors the types express. But I'm going to confine myself to how the cell types within the retinal ganglion cell class diversify. And by way of background, let me review some key points that have been discovered about the lineage of cell classes. And by classes, I mean the ganglion cells, bipolar cells, amacrine cells, and so forth. And there's sort of three, I would say, big tenets that have been worked out over the years. First is that all of these classes arise from multipotential progenitors that divide and give rise progressively, let's say, to a retinal ganglion cell, maybe later a amacrine cell, maybe later a rod, and so forth. And that was discovered in 3 labs, more or less at the same time. Christine Holt and Bill Harris, your colleagues, Scott Fraser, and Connie Setko and her colleagues. Second big idea is that as the progenitor goes through its cell divisions, its propensity to generate different types of cells, first retinal ganglion cells, last rods, and so forth, changes because the intrinsic competence of those progenitors to generate a particular cell type changes. It's not entirely clear how much that change is driven by intrinsic timekeeping mechanisms or by extrinsic factors, but Connie Sepko, I'd say preeminently, has got a lot of evidence for the idea. And then the third big idea is Bill Harris's, which is that the competence is not a determinative one, but rather a stochastic one. That is, at any given point, the probability that a progenitor will spit off a, let's say, ganglion cell or amacrine cell is a matter of probabilities. It's not an all or none phenomenon. Now I'm going through all this because when we get to cell types, that is how a retinal ganglion cell diversifies into its 46 types, a newborn amacrine cell into 63 types and so forth, we know nothing about any of these. That's largely because when Sepko, Harris, and all were doing their work, the number of types really wasn't appreciated and the markers to distinguish multiple types were not available. So we thought we'd have a crack at it. And what we did to begin with, and the main we here is Karthik Shekhar, a computational biologist, and Irene Whitney, a neurobiologist. Karthik now has his own lab at UC Berkeley. And so we began by asking about two extreme models. One extreme is that there are 46 different types of progenitors, each one of which is fated to give one type of retinal ganglion cells. And the other is that it all happens postmitotically with some kind of stepwise determination in which a multipotential precursor postmitotically gradually becomes committed to a particular fate. And so to distinguish that, we repeated our isolation of ganglion cells and transcriptomic characterizations at earlier and earlier times, all the way back to when the first ganglion cells were being generated. And what we found was that the number of clusters increases. And although I'm not going to talk about it so much, the clusters became more clustery. They became better separated from each other. And so at face value, with a lot of controls that I'm not going to go into, that kind of argues against a strictly predetermined scheme. That is, there's no reason at all to believe that when ganglion cells are newly postmitotic, there are 46 types of them. So then we asked about the stepwise mechanism. And again, you can imagine two extremes. One is that each postmitotic ganglion cell, I'll call it a precursor, is committed to give rise to a small number of types, and that plays out. The other is that they remain uncommitted early on. And so any given precursor can give rise to some of the same types that a different type of precursor gives rise to. And sort of the computational way to have a first crack at this is to organize what's called a confusion matrix, in which one takes an early type and asks for its transcriptomic similarity to later, more numerous types. And so in a specific model on the left, a given early type would be transcriptomically similar to a discrete set of later types, whereas in a completely non-specific model, the transcriptomic relationship would be more or less random because anything can become anything. And the data are here, and you can see it's sort of somewhere in between. But clearly, it's not the case that early clusters seem to be fated by this criterion, which is admittedly indirect, to become specific groups of adult clusters. So we feel that we can tentatively rule out a completely specified model. But that, of course, is a completely specified model at the level of these clusters or types. And so it could be different from the point of view of individual precursor cells. And I'll give you, again, two extremes. One would be that the precursors in an individual cluster are actually each one fated to give rise to a particular type, but they're so transcriptomically similar at this early time that we can't tell them apart. The alternative is that within a cluster, individual ganglion cells truly are multipotential, truly uncommitted. So there is a way to distinguish these alternatives, and that is to visualize a precursor, follow it through to adulthood, see what type of cell it becomes, and then run back time and repeat the process over and over again. But that won't work. That violates the laws of physics. And so Karthik decided to use an indirect computational method called optimal transport, which predicts how a cell at a given time distributes its so-called transcriptomic mass to cells at a later time. And again, I don't have time to explain this, and to be honest, I'm not sure I could do a good job of it anyways. But the key point here is that it is dealing with cells one cell at a time, independent of the fact that they're clusters. So they're not really looking at the possibility. It won't get caught by the possibility that we're mistaking types within a cluster for being homogeneous. And the upshot is that indeed, by this criterion, the cells are very multipotential. Each one contributes its transcriptomic mass, which we view as a proxy for fate, to an average of 10 to 12 adult types at the earliest stages. And then that multipotentiality decreases until in adulthood, of course, it's one, because every cell has become what it's going to become. And the UMAPs, which are like Tisne's on the right, are just to show that this multipotentiality is shared by many, many different clusters or cell types. It's not just a property of a few. So that then makes us believe that these precursors are truly multipotential early on, each one having different probabilities of turning into different sets of adult types, and those types are, of course, overlapping one precursor to another. So, of course, now the big deal, and we haven't done enough here, is to ask what are the types that an individual precursor can give rise to. And I'll give you just one small result. These are optimal transport results displayed in the form of what's called a Sankey plot, in which one looks at the weight in which cells at a given early time give to the 46 adult types, which are shown at the right, with every intermediate point in between. And what you can see is that some groups of cells, for example, those that are characterized by expressing the transcription factor TBR2 in adulthood, or expressing NeuroD2 in adulthood, seem to have some coherence in which groups of early cells they derive from, whereas others, like those expressing the transcription factor FOXP2, don't. And so what we think overall, and I should point out for those of you into the details, TBR2 marks intrinsically photosensitive retinal ganglion cells, but not only them, some other types that also turn out to be pretty close relatives. So what we come up with then is a tentative model of diversification in which the postmitotic cells are initially multipotential. They're biased towards generating subclasses, and they separate. It was kind of inherent in the Sankey plots, but I didn't point it out, by an asynchronous process of fate decoupling, where some become committed to a particular fate earlier than others. And I put these words in blue because it's kind of interesting to me, at least, that they resemble in many ways the big ideas that Harris, Fraser, and Sepko came up with for the diversification of cell classes. It's just in this case, the multipotentiality, stochastic bias, and asynchrony are occurring postmitotically rather than during the process of mitosis. So let me turn now to the second story, and this is about vulnerability of ganglion cells. And it's known very well that in many neurodegenerative diseases, the insult is shared by many cell types. Think of ALS, Parkinson's, Alzheimer's, but only certain cell types are vulnerable and end up dying. And so we thought we might be able to look at this for retinal ganglion cells, because in mouse, after you crush the optic nerve just outside the orbit, 80% of the ganglion cells die in 2 weeks, 90% in 4 weeks. And then I'll come back to it in a minute. Almost none regenerate axons, but a few can be induced to do so. And so in a collaboration with Zhigong He at Children's Hospital, we initially asked, which 80% die? Is it completely stochastic, or maybe the cells that were active when the nerve were crushed, or were inactive, or were near a blood vessel, or had a little bit more trophic factor? Or could it be that at the limit, 80% of cell types are completely vulnerable and they all die, whereas 20% of cell types are completely resistant and they all survive? And so we asked that initially using a small set of transgenic lines. And the answer was that there is a big cell type bias. Alpha retinal ganglion cells and one of the intrinsically photosensitive ganglion cells, for example, survive extremely well, whereas many other types fail. TUJ1 is the marker in this case for all ganglion cells, and you're seeing the overall loss of 80 to 90%. So once we had the atlas, we thought we could go back and do a more comprehensive look at resilience and vulnerability, and also characterize the resilient and vulnerable types. The idea being that if we found some genes expressed by resilient types, would it be possible to stick them into vulnerable types and make them more resilient? Or could we knock out genes in vulnerable types and make them more resilient? So what we did was look at the frequency of the 46 types in the atlas, and then crush the optic nerve, wait various times thereafter, isolate the ganglion cells, and do the same thing over again, and then divide one by the other to give us a measurement of resilience. And what you can see are a few things. First, there are really dramatic differences. The most resilient types survive to the tune of 80 or 90%. The most vulnerable types die to the tune of more than 95%. The other thing we found out is that it's a continuum, not a dichotomy of resilient and vulnerable types, which was a point that was sort of left unsettled when we only could look at a few types. So now we could go into these types and ask whether they're genes selectively expressed by resilient or vulnerable ones. And we could do that in intact retina or at various times after performing optic nerve crush between a half day and 3 weeks. And the bad news is that we couldn't find any genes that were either expressed by all and only resilient types or all and only susceptible types, or whose expression was graded with resilience and vulnerability. But clearly there were some interesting possibilities, and so we went on and tested a few of them. And the we here, I should say, is Nick Tran, who now has his own lab at Baylor, Karthik Shekhar again, Anne Jacoby, and Irene Whitney. And so here are two that were particularly interesting. Urocortin is a peptide that's upregulated in some resilient cells, but no susceptible cells. And its secreted intrinsic inhibitor, CRHBP, a binding protein, is selectively expressed in many susceptible but no resilient cells. And that's true before or after crush. So the idea is urocortin is a peptide in a family with CRH, and CRH binding protein is secreted, binds urocortin and CRH, and prevents both of them from activating the receptor. So this seemed like an interesting conjunction. And so we went on and either overexpressed urocortin or injected urocortin protein or knocked out CRHBP using a CRISPR-based approach. And in all those cases, we significantly increased the survival of cells at 2 weeks after nerve crush. The control value is shown by the red horizontal line, with pink being the error bars. And so this is not a huge effect, to be sure, but it's very encouraging of the approach. So I'm going to skip this and then go on to what we're doing now. And in the new step that Anne Jacoby is leading with Nick and now Wenjun Yan, we're trying to look harder at how resilience and vulnerability come about by looking at interventions that can save some of the ganglion cells and actually promote axonal regeneration from some. And the three interventions we're using are ones that Zhigong, still our collaborator, had discovered many years ago. And so they've been validated in multiple studies in his lab and other labs. And they are deleting a phosphatase called P10, which is an endogenous inhibitor of mTOR signaling, deleting a transcription factor called SOX3, which is an intrinsic inhibitor of JAK-STAT signaling, and overexpressing the neurotrophic factor CNTF. And we did it in combinations, either P10 conditional knockout alone. It's being knocked out only in ganglion cells to avoid indirect effects and for P10 lethality. P10 knockout with CNTF or all three interventions. And you can see here, really confirming what Zhigong and others had shown, that they substantially and in a graded fashion increase survival of ganglion cells and the regeneration of axons from a minority of those cells that survive. So the idea now is, again, at various times after performing these interventions in optic nerve crush, to have a look at which cell types survive, which cell types regenerate, and what genes they're expressing. And so for survival, the answer is a fairly simple one. It turns out that all of these interventions, with only a few exceptions, promote the survival of cells in proportion to the fraction that survived initially. So they're still resilient and vulnerable types, but whatever the vulnerability was after crush alone, superimposed on that is an overall increase in survival. For regeneration, it was a little more complicated because what we needed to do was separate those cells that regenerate in a given retina from the much larger number that survived but didn't regenerate. And so to take that on, Anne Jacoby devised a really beautiful and extremely difficult surgical method in which she injects a fluorescent dye, microruby, into the optic nerve, far enough away from the nerve crush that it won't be taken up by stumps, and close enough to the nerve crush that it can intercept a fair number of the regenerating axons. And she can then go back and look in the retina at the cells that are microruby positive, and you can see them on the right in a whole mount of the retina. Those are those little orange dots. And then all the cells in this experiment are ganglion cells, are labeled with YFP. And so she can then use FACS sorting to collect the cells that are yellow, that survived but didn't regenerate, or yellow plus red that survived and regenerated. And obviously, difficult method, lots of controls that I won't go into. So here's the result. And it was a little more complicated when we only knock out P10. Although many types of cells survive, the regenerators are actually predominantly a single small subclass called alpha retinal ganglion cells. This turns out not to be a surprise because we'd actually shown that already in an earlier study. What was a little more of a surprise is that when we look at all the interventions, the cells are regenerating in rough proportion to their survival. So what seems to be the case is when you activate JAK-STAT signaling, which both SOX3 knockout and CNTF overexpression do, you overcome the type-specific limitations on regeneration imposed by P10. And so, of course, it's particularly interesting to find out what's going on downstream. How are these interventions managing to cause regeneration? I should point out, by the way, that P10 and SOX3 are not great therapeutic candidates on their own because they're both tumor suppressors, so that would be bad news. And CNTF has been tried, but isn't terribly effective all on its own. So Anne could now go in and ask, what are the genes that are expressed selectively by regenerating cells compared to those that survived but didn't regenerate. And using the same sort of strategy we used before and that I described briefly, she's been able to test them by overexpression of 3 genes, the transcription factor WT1 and 2 neuropeptides, CRH and gallinan, by overexpressing them. And indeed, all 3 of them promote regeneration of axons, as you can see in examples on the left bottom, and the group statistically analyzed data on the right. And again, this is not enough to restore vision. So far, no intervention that anybody's used has been enough to restore useful vision in a mouse. But it certainly, again, encourages our thought that by figuring out how these interventions work, we may be able to move towards getting more useful ones. Now, what we'd really like to do, though, I mean, that's the long-term goal, but in the short term, we'd like to have a deeper look into the gene expression programs that are regulated by PTEN, SOX3, and CNTF, so that we can sort of have a more comprehensive idea of what they're doing and how they're doing it. And so what Ann, Nick, and Wenjun have done is to take all the cells from all the interventions at all the times and essentially recluster them, and then use a variety of computational methods to figure out what genes are expressed and how those genes vary by intervention, by time, by survival, by regeneration, and so forth. And the key point here is that 4 different methods all end up giving us 3 gene expression modules, that is, genes expressed by sets of cells. They're not identical. And from one point of view, it's not a big shock because, of course, all 4 of the methods begin with the same data set. But they all make very different assumptions and use very different algorithms to come up with their results. So one is to cluster the cells by type of intervention. One is to cluster them all by transcriptomic similarity without any regard to what type they are. One emphasizes the states over the types. And one, perhaps the most interesting, is called scenic, which digs into the data to come up with what are called regulons, that is, groups of expressed transcription factors and co-expressed genes that are predicted to be transcriptional targets of those factors. And again, we don't know enough for me to give you sort of a comprehensive view of the result, but I'll just give you one example here of 3 different modules, in this case from Seurat. In this case from scenic. And what you can see on the right are in blue the cells that end up being in each module. And you can see that they're not completely, but largely non-overlapping cells. And on the right, although you can't really read it, is some indication of what some of the key genes are. But basically, there's one module that seems to be expressed largely by vulnerable cells that are either destined to die or already dying. And the genes expressed in those cells include many that have been already implicated in apoptosis, including transcription factor ATF4 that's been implicated in apoptosis. Another module is expressed by cells that are surviving but not necessarily regenerating. And that has, interestingly, a lot of genes in it that have been implicated in neural development. One can sort of make up the story in which these cells are trying to survive. They're sort of regressing to an early developmental state, but they're not quite making it. And then the third module, selectively expressed by the regenerating cells that include the candidates that I already showed you, have a lot of genes that are already known to be implicated in axonal regeneration, so-called RAG genes, regeneration-associated genes in other systems. And so, you know, we're now digging in. I'm not going to stop and talk about this to really look at what some of the genes are, what they're where they're expressed, what the regulatory networks are of them. So let me come to the last and shortest story. Shortest because we frankly don't have a lot of results, but I'm going to tell you a little bit about it in hopes of provoking discussion, because to be honest, we're in need of ideas. And so the idea here is to see if we can use the retina to find out something about the evolution of cell types and classes, kind of by analogy to what people already know about the evolution of genes and gene families. That's a very well-developed field. And there should be, but there isn't, a corresponding field for cells. And we think the retina is great for this enterprise because, as you probably know, its overall structure, its layers and cell classes are beautifully conserved amongst all vertebrates. And you can't say that for other structures, for the cortex. It's a little hard to look at the evolution of cell types in the cortex because there are whole parts of the cortex that are missing, for example, in birds or fish. And in some of them, the whole cortex is missing, frankly. So what I've told you about so far is a cell atlas from the mouse. Now, a few years ago, Yirong Peng began expanding this search to look at primates, first macaques and then marmosets and humans. And her reason at first was that although the mouse retina is quite a wonderful model for studying the development and function of neural circuits, it actually is a horrible model for looking at vision. And that's because our vision is dominated by a central fovea or macula that's only a small fraction of the whole retina, but accounts for essentially all of our high acuity vision and really most of our color vision in daylight illumination. It's clinically critically important because diseases like age-related macular degeneration, which is probably the #1 cause of blindness in the first world, selectively affect the macula. And among mammals, only primates have a macula. So these are structures and functions and diseases that really cannot be adequately studied in mice. So what Yirong did, starting with macaque, was separate the fovea, which is the central bit of the macula, from peripheral retina, and look at the transcriptomes of the cells, coming up with atlases of, as I said, first macaque, then human, and then marmoset, separately for fovea and periphery. And of course, when she had those atlases, she could then ask, what is it about the fovea that makes it different from the periphery? Different types of cells, different proportions of shared types, similar proportions of shared types with differentially expressed genes. That's published. I'm not going to talk about it, but the short answer is that all three things are true, but the vast majority of types are conserved. So that's really not it. But every single type has a substantial number of differentially expressed genes between foveal and peripheral cohorts. And so we expect that in those differentially expressed genes are going to be the ones that give foveal cells their distinct structures, their distinct functions, and their distinct connectivity. At any rate, we could then begin comparing the atlases of the primates to the mouse. And in the past few years, we've expanded that to look at a number of other mammals, rabbits, Peromyscus, which is a wild mouse, squirrels, pigs, ferrets, basically anything that somebody is euthanizing in a lab for another purpose but can spare the eyes. And we've also done some work, some of it published, on chick and zebrafish, and are beginning to work on lizards. And here's just sort of a not very, but fairly recent census of the number of types that we're finding in these species. And so what we're really going to focus on now are the bipolar and the retinal ganglion cells. So I'll finish with just two snapshots. One is about cell classes. And this is a comparison that Wenjun Yan made of two primates, human and macaque, mouse and chicks, looking at classes. And what she did, kind of what I showed you about the interventions, is take all the cells together and recluster them. And you can see at the top the clusters by class. In this case, we're not going deeper to the types. And on the bottom, if you look, the 4 colors represent the 4 species that contributed to this map. And what I want you to see is that for every class or subclass, cells from all 4 species contributed. So in other words, we knew that at a structural and functional level, the retina was highly conserved amongst rodents, primates, and birds, but we didn't know whether that was also true at a molecular level. And the answer is, it is transcriptomically. All of the cones are each other's relatives. All of the rods are each other's relatives and closest relatives to the cones, glycinergic and GABAergic amacrins and so forth. One surprise out of this for aficionados in the audience is that a really fascinating type of amacrine cells, the starburst amacrins, are actually more closely related to retinal ganglion cells than they are to the other amacrine cells, sort of driving home their special status that people have already commented on by other criteria. For types, we know less, but here's just 2 examples. So when Wenjun went on now and reclustered amacrine cells by type, or reclustered retinal ganglion cells by type, she got quite different answers. And I think you can see that intuitively on the left, where the individual amacrine cell clusters almost all contain cells by the particular criterion from all 4 species. And the average number of species that contributed to each cluster was 3, again by this criterion. Similar for bipolar and amacrine cells. Whereas for retinal ganglion cells, they seem to cluster separately. And again, intuitively, you can see that. There's some purple clusters, some red clusters, some green clusters, not so many rainbow clusters. And so on average, only about 1 species contributed to each of these clusters. And so this kind of supports the idea that the outer retina may be a beautifully conserved and optimally wired graphics card that is conserved through a lot of vertebrate evolution, whereas a main site at which evolution acts to essentially suit the visual capabilities of each species to its ecological niche is at the level of retinal ganglion cells, where the information is being conveyed from the eye to the rest of the brain. And just last month, Karthik Shekhar came up with another intriguing piece of evidence that supports this idea. And this is going to be the last slide I show you. So if you look at the species, this is not all of them, but most of the species we've profiled. On the left, you see a conventional evolutionary map of their relationships. Human and macaque and marmoset, of course, are each other's closest relatives. Mouse and Peromyscus, squirrels are pretty closely related. Chicks and fish are outgroups. If we look now at the transcriptomic relationship of the bipolar cells, it fits that evolutionary relationship perfectly well. On the other hand, everything goes haywire when you look at ganglion cells. When you look at the transcriptomic relationship of ganglion cells, zebrafish are the closest relative of the primates. Pigs and squirrels are close relatives, which they shouldn't be. Chickens are the closest relatives of the rodents. And we're not really sure what this means, but one simple idea that certainly intrigues us is that maybe a lot of evolution is occurring more or less by neutral drift. So the separation among species is in some sense a clock of how long it's been since they diverged. And that may be true for bipolar cells. They're evolving by neutral drift, just as is the whole species. Whereas for retinal ganglion cells, there are selective pressures, whether they be purifying selection or whatever, that those selective pressures are acting and they're overriding the evolutionary clock for reasons of their own. Again, the idea would be that it's at the level of ganglion cells that evolutionary pressures are being felt the strongest to make sure that the right kind of information is being transmitted from the eye to the rest of the brain. And of course, this is evolution, so we're never going to be able to test it, but we're trying to get more species to see if this holds true and see if we can do more computational tests and look at what types there are that, for example, make zebrafish look like macaque in this respect. So this has been the most collaborative project I've ever been involved in. It's been a real pleasure. I mentioned Steve McCarroll and Zhigong He as I went along. Mike Doe was a huge help in the initial studies of primates. Aviv Ragev got us going on all the computation. In fact, Karthik Shekhar began as a postdoc in her laboratory. And the zebrafish work was a wonderful collaboration with Yvonne Kelch, a graduate student in Herbert Bayer's lab. So I'll stop now. Thank you very much.
Okay, great. Thank you, Josh. It was a great talk, really interesting. And now we have a couple of questions in the chat that I will read to you. So first we have from Professor Marla B. Feller. She asked, how do you think neuropeptide overexpression could enhance survival? Are there photoreceptors on these cells and it somehow promotes signaling in the same cells that secrete them?
Yeah, so we don't know. First of all, I should say thank you, Marla, for getting up so early. In some cases, in other cell types, DRGs and so forth, it looks like one main way they act is by increasing cyclic AMP levels and activating the whole pathway downstream of cyclic AMP. We don't know at all whether that's the case here. In terms of receptors, yes. For urocortin and CRH, for example, the CRH receptor is expressed by pretty much all ganglion cell types. We know less about the galanin receptor.
Okay, thank you. Then we have George Cafetsis. In what light conditions were mice kept post-ONC? Could neuronal activity play a role in the survival of regeneration processes?
Yeah, that's a great question. There certainly has been some evidence, not from our lab, but from others, that activity can promote survival of ganglion cells. That started first, I think, with Ben Barris' demonstration in cell culture, and there's some evidence in vivo. We kept them in a normal daylight cycle. We haven't tried changing activity in this context. We did in another context. If you give me another 30 seconds, we have been interested in whether the maturation of ganglion cells is affected by activity. And so we've done a bunch of dark rearing experiments, and they have, to me, shockingly modest effects on the transcriptome of adult retinal ganglion cells.
Okay, thank you. Then I have a question. So I observed that there are so many different types of amacrine cells in the clustering you said, and I know that they have different morphologies. So I was wondering if you found many more pathways that are different among them, or very different genes among them, more than in retinal ganglion cell or in bipolar cells.
Yeah, there's more types. And certainly when you look at them transcriptomically, the GABAergic amacrines and the glycinergic amacrines sort into different clades. Our attempts to try to come up with further subclasses, the way we've done with ganglion cells, have not really been very successful so far. So my guess is they're there, and we just haven't done a good enough job of looking for them.
Okay. Now we have a question from Leon Lagnado. How far can we relate transcriptomic overlap divergence of cell types between species to different in visual processing within some species, within those species?
Yeah, that is the question. That is what Karthik is really focused on, and we just don't know. There's nothing I could say that wouldn't be stupid, to be honest, so I won't say anything.
Yeah, well. Okay, so we have another question from Simon Laughlin. Great talk. Slightly surprised that Owen bipolar cell cluster with off, given ideas that they evolved from photoreceptors.
The bipolar cells are related to photoreceptors, you're saying? Yes, I think they are pretty closely related to photoreceptors, and I think, I mean, that seems reasonable to me. They both have ribbon synapses, and they have a lot of physiological features in common. So you know, I'm not sure what to say. I don't think anything's expected, but I think that transcriptomically, that amacrines and ganglion cells are more closely related to each other, and photoreceptors and bipolar cells are more closely related to each other. And we've tried to look a little bit at what's known about the most primitive retinas before there were a lot of cell classes. And I guess the best I can say is what we've seen is consistent with that, but not really strong evidence.
Okay. I think there is a last question. Is there some way to identify the genes that are more susceptible to evolution?
Yeah, again, in this very, very preliminary little story that I told you, that has not been done yet, but certainly there are methods that people, hi Marla, there are methods that people have worked out to track the evolution of genes. And in fact, that's a pretty well-established field. And so Karthik does believe that he'll be able to dig in and do that, but again, it hasn't been done yet. You know, whether it can be done with enough certainty to be useful, I think remains unclear.
Okay, thank you, Josh. So yeah, I think we'll finish the talk now. Thank you, Josh, and thank you everyone for being here. And now I will leave the transmission on for a while so that people can join. But yes, please join the discussion now. Thank you. Okay, so we have more people joining.
Josh, so good to see you. It's been a long time.
It's good to see you. I only hear about you from Karthik these days.
I know. I talk to Karthik all the time. He's doing great. I hope he's doing great. It's stressful.
I think he's doing great. He was in Boston a couple of weeks ago for some chemical engineering method. You got to get him out of that department. But,
yeah. It's a good department, I think, isn't it? Well, anyway, we could talk about this.
No, I think it's a great department. They just have to teach a bunch.
Yeah, he has to teach a bunch, but he really kind of likes to teach, or so he told me. But anyway.
Yeah, I like teach by grab, but not that much of it.
Yeah. Well, it's a little bit, you know, those hard money positions. You got to get paid for doing something. So, but I do talk to him a lot about this. So, I have a list of questions I'm going to go run and get. Then I'll let other people ask questions. So I'll be right back.
Josh, could I ask a sort of general question?
Sure.
How do you, I'm thinking a lot about homology versus analogy in evolution, and where you see this finding that the retinal ganglion cells are somehow more related between fish and primates, for instance, than to other species. Is that because they're more analogous, or?
Yeah, this is the downside of talking about data that we don't understand. I mean, I like the idea, and I credit Karthik for this, that the wacky transcriptomic relationships are a sign that there was selective pressure exerted on the types of ganglion cells that overrode the dominant feature of the other cell classes, which is essentially a timing mechanism dominated by drift.
Yeah.
But then what you're asking is why? What is that pressure? Can we learn something about that? What the pressure is from, say, the fact that fish and primates are related to each other? And I just have no idea. I really have no idea.
My first thought was that it must have something to do with color vision,